We had already discussed the first part of photosynthesis, which involves the light reactions. That was talking about how we actually capture the electromagnetic radiation that emanates from the sun. Those sun rays, the photons, are collected by the chlorophyll molecules as well as other pigments in chloroplasts, sent to the reaction center where the water gets split, where the electrons from water get highly energized, and those electrons are pumped not pumped, they are transferred from one complex to another. They start in photosystem 2, pass through plastoquinone to the first pump that pumps protons into the thylakoid space, to the plastocyanin, and end up back at the photosystem 1, where additional energy is collected and transferred to those lower energy electrons now. And the final electron acceptor in this pathway was NADP+. So an NADP+ reductase makes NADPH. And so what we've actually done in the light cycle is to generate a proton gradient across the thylakoid membrane with a lower pH and more protons inside the thylakoid as compared to the stroma. And we've generated some NADPH, which is nothing more than an electron carrier that can be used in the electron transport chain of mitochondria to generate ATP. Okay, the last part of the light cycle was there is an ATP synthase in the membrane of the thylakoid, and the protons flow down their electrochemical gradient out into the stroma, generating the energy to turn the turbine, which allows for the ADP and PI, the substrates, to change their conformation enough to make a phosphoanhydride bond. So that leaves us with some ATP, and the idea is now, what do we do with that ATP? In the dark cycle can occur in the absence of light, so this can occur at night. Okay, it'll occur until all the ATP is used up. But what it does is it fixes carbon, and the carbon comes from carbon dioxide, and it fixes carbon into a carbohydrate molecule. It's called the Calvin cycle because Calvin was the guy who originally identified this cycle, and we're going to talk about the cycle in very broad terms. This is an energetically unfavorable reaction because we're going from disorder, a bunch of CO2s are going to be used, to more order. You're generating a carbohydrate molecule, a 6-carbon sugar. Okay, so just remember what happens in the light cycle. We break water, we collect light, we collect the energy from light, we make ATP and NADPH. NADPH is ultimately going to be used to make ATP, and water is generated, oxygen is generated in the reaction when we split water, and that's just a waste product for the plant, but we obviously need that to live. Okay, the overall goal of the dark cycle is to take those products and use them to generate sugars. So the first step of the dark cycle is probably the most important step, and it involves the carbon dioxide, so a CO2 molecule, is attached to another molecule called ribulose 1,5-bisphosphate. Okay, and they're shown here in this diagram. And in the diagram you see ribulose is a 5-carbon sugar. It already has 2 phosphate groups attached to it. And the enzyme that does this reaction is called ribulose bisphosphate carboxylase. So we're adding a carbon onto ribulose bisphosphate, ribulose 1,5-bisphosphate. The enzyme has been given the nickname Rubisco, ribulose bisphosphate carboxylase, and it is the most abundant protein on Earth. And you could think of, why might that be? Why would you need so much of this protein? Well, this is happening all the time anywhere that the dark cycle is occurring. So any organism that utilizes photosynthesis, plants, bacteria, utilize rubisco. And what's interesting about this is it makes up approximately 50% of the protein that's in a chloroplast. That's a lot of protein. Okay, this is an enzyme. And what do you remember about enzymes? Enzymes lower the activation energy of a reaction by changing the conformation of the substrates to allow for that reaction to occur more quickly. Does the enzyme get used up? No. Does the enzyme eventually get used up? Yes. Enzymes, since they're mostly proteins, will, over time, become degraded. But in this case, what we're talking about is rubisco, the enzyme, allows for the addition of a carbon to ribulose 1,5-bisphosphate. It goes into this intermediate, and it immediately, the 6-carbon molecule that gets made is immediately cleaved into 2 3-carbon molecules called 3-phosphoglycerate. Sometimes we just call it 3-PG. Okay? So that is an incredibly important reaction because we're using energy to do this. Okay? What they're not showing you is this requires a lot of energy for this reaction to occur. And that's because we're making a covalent bond. So let's get back to why is this the most abundant protein on Earth? One really interesting thing is that rubisco is an incredibly inefficient enzyme. It does not do this really well. And so we need tons and tons and tons of this enzyme to do this reaction over and over and over again. And you might ask, well, why is it so inefficient? Why doesn't it become more efficient? Do you think it's become more efficient over time? Probably yes, actually. But right, over the course of evolution, it's worked. And if something works, the only way for it to get better at doing that is through random mutations, and the random mutants that are better will survive better and cause there to be more offspring of those mutants, and apparently it hasn't mutated fast enough to be that much better. It's still a very poor enzyme. Okay, so far what we've done is we've taken a 5-carbon sugar, a ribulose bisphosphate, we've added 1 carbon to it, and that 6-carbon sugar has now been split into 2 3-phosphoglycerates. So there's a lot of numbers involved in this, so you see the reaction here, 3 carbon dioxides plus 9 ATP, plus 6 NADPH plus water leads to 3 glyceraldehyde 3-phosphate. So that's a lot of energy we're putting in to generate glyceraldehyde 3-phosphate. What happens to the glyceraldehyde 3-phosphate? Glyceraldehyde 3-phosphate actually is an intermediate in glycolysis, so it's not that this reaction is happening all by itself. It happens elsewhere in cells, but we actually take that glyceraldehyde, we, being plants, okay, now I'm acting like I'm a plant, gets exported into the cytoplasm where ultimately it gets made into glucose or fructose or some sort of sucrose molecule. So any of this glyceraldehyde 3-phosphate that's excess in the stroma of cells will be converted into starch. So these are the ultimate reactions I would like you to know, the light cycle and the dark cycle. What happens in each? What are the products of each? Okay, glyceraldehyde 3-phosphate, all right, is the ultimate product that gets exported and made into glucose. So the bottom line is we're taking water and CO2 in the presence of energy, light from the sun, and we can make sugars and O2 as a byproduct. I've cut this really short because there are multiple stages in the dark cycle, and if you take biochem, you're going to learn each of those different steps, but what goes in, what comes out is what I really want you to learn. We began talking about this, so chloroplasts are one type of plastid, and we have multiple different kinds of plastids, and they all come from something called a proplastid. So a proplastid is, okay, here's a plastid, but it doesn't know what it's going to become. It could become this kind of plastid, or it could become this kind of plastid. And what's interesting about that is it depends on the differentiation of those plastids, depends on the expression of genes that are in the cell. And so light makes one kind of plastid into a chloroplast, but other plastids may never see light. And where that occurs is in roots of plants or tubers, all right? And tubers are basically like a potato is a tuber. And so they never see light, and they are called leucoplasts. They're actually white. If you think about what a potato generally looks like, right? It's a white growth underground that allows for the storage of sugars or carbohydrates. And right, we eat them because they taste good. So different kinds of plastids will have different kinds of functions. The chart on the left here shows different kinds of plastids. They're all emanating from a proplastid. Okay, the proplastid can turn into a leucoplast or a chloroplast or a chromoplast. And the chloroplasts and the chromoplasts have color associated with them. All of the leucoplasts are storage molecules, and so there's clever names. Proteinoplast, what do you think that stores? It stores proteins. Elioplast, I don't even know what that stores. A myeloplast. All right, these are different kinds of storage devices for different types of molecules. Okay, all of this is really storing energy in a form that we can utilize in another way, either through eating it or using it downstream of the plant itself. So that finishes the end of the Calvin cycle, and I'm going to play a little video here that I hope you watch and look at and really understand how this is all working in a plant cell.
The Calvin cycle is a series of reactions that results in conversion of carbon dioxide into the organic molecules needed to build new cells. It occurs in the stroma, which is the area of the chloroplast surrounding the thylakoid membrane. During the Calvin cycle, carbon dioxide, CO2, is added to a 5-carbon molecule called RUBP. The resulting 6-carbon molecule is unstable and quickly splits into 2 3-carbon molecules, called 3-phosphoglycerate. Using energy from ATP and reducing power from NADPH, which are products of the light reactions, the pair of 3-phosphoglycerates move through a series of reactions and are converted into 2 molecules of glyceraldehyde 3-phosphate. When several of these glyceraldehyde 3-phosphate molecules have been produced, some combine to make glucose, while others are reused in the Calvin cycle. To generate an entire new glucose molecule, the cycle has to turn several times, because each turn of the cycle adds only 1 carbon atom from each molecule of carbon dioxide.